Okay. Oops, what I did. Okay, so thanks, Clarice, again for this amazing symposium, and thank you all of you to be here today to watch my presentation. And I'm trying to be real quickly so we can all head over to our gala dinner. So the topic of my talk today is about how to use biogeographic analysis to try to evaluate or understand how phylogenetic reticulation happen on, especially on the deep time. More specifically, I will deal with a group of gingers that my colleague Eliska already talked about today. So I'm pretty sure that everyone here knows how important our phylogenies are for understand the relationship of species, family orders, you name it. But the issue here, as we are already seeing, is that sometimes this classically bifurcating graph that we call phylogenies are not always true, or at least for some clades, they cannot be truly true. And under microevolutionary points of view, studies with populations already noticed the hybridization and gene flow is a very common phenomena within populations or within closed species. But in a macroevolutionary approach, this hybridization is not so easy to capture. But now actually with more and more, we are discovering that ancient reticulation actually has profound roles in the origin of different clades, spanning from entire orders to single species, also spanning through hundreds of millions of years to more recent events like in the last glacial maximum, the last 20,000 years. Also, deep time reticulations can indicate clade diversification, as Clarice showed us on this morning. So these reticulations actually could not only be dead ends, but also could improve the exploration of new niches for these particular clades. So, for example, here I have two examples of reticulation that are already published. One is Ericaeus, that is a hybrid orange between the ancestrals of Cornelis and Gentianides. This was super old, 125 million years old. While here, in the other hand, we have the common walnut, Jungla regia, which is a hybrid between two ancestral relative of other Junglans. So this is only 3.5 million years. So you can see that reticulation actually has a profound role on the diversification of different clades, here orders and species. But of course, try to find these reticulations in a macroevolutionary point of view is sometimes hard because for sure we need to have a lot of data to try to find this kind of stuff. But also it's difficult to test under in an analytic point of view because reticulations can be sometimes blurred with incomplete lineage sorting. So they are both events that shows similar patterns on phylogenetic. So again, microevolutionary studies already noticed, and they are using like several models and simulations to see how the things happened in the past. But again, this is in the microevolutionary point of view. When we are moving back to the past on the microevolutionary, trying to deal with families, orders, and you name it, this could be a nightmare, like for computers to try to analyze this kind of stuff. Sometimes it's quite impossible. So there are many parameters like population size, migration, when the splitting time of this. So to deal with this kind of amount of data, luckily now we are some techniques like network theory was implemented to use to try to understand how reticulation happened in the past. And here I'm pointing out two really good tools for like Phylonet works. Both of them are deals with different way of how to estimate these reticulations, but the issue here, um, sorry, the issue here is that even on these particular tools, the reticulations can produce some spurious reticulations. So that doesn't make sense in an empirical point of view. There is no biological meaning because we need to use a reduced subsample, and also sometimes the reticulations is between two clades that doesn't occur in the same area. So what we should do actually? So but if you think about it, how reticulation could have happened in the past, you think that actually reticulation had happened in the past, the ancestral lineages must have overlapped locally and temporally. So they must meet at some time and at some place. So in this hypothetical example, I can show you here that for this lineage A and B produce this hybrid X, both of these prerogatives must be met. And so both of them must occur in the same area or in a neighbor area, or some migration must have happened in a common area. And also this must have happened in the similar time period. You must, because if you have in the same area, but they are occurring in a different time, it doesn't make sense because they are not gonna meet to produce these hybrid reticulations. And to test this framework that reticulation must match these two prerogatives, we select the genus Curcuma, which is a very economically important genus in Southeast Asia, around 140 species. Probably you already know Curcuma longa, which is like the turmeric, probably like the yellow powder that you put on your curry. And from our previous work on our group in Prague and other places, we, especially Eliška, already found that the genus Curcuma, the genus Curcuma has these 5 lineages. So the subgenus Curcuma, which is here in the circles, are around everywhere here in West India, also here in Northeast India, Bangladesh, and some parts of Myanmar, while the other lineages like Ecomata, Hicheniopsis, Pierana, and Rusciana are more centered here in Indochina, like Myanmar, Bangladesh, Thailand, and Vietnam. So we also select this. We also select this group because we already have some works like Eliška showed you that we have reticulations happen here in the past. Actually, the entire subgenus Curcuma is a hybrid origin. So you can see from the red arrows. We also found some other reticulation events within the subgenus Curcuma, but today I'm gonna focus on this one that gave birth to the entire subgenus. Is also not worth to show here is that Curcuma is the most specious subgenus on the entire genus, and the one is subgenus Curcuma is also the one that has the most different niche viability. So they occur here all the way here in the West India, also here in Sri Lanka, some parts are here for Northeast India, while the others subgenus only occurs here in Indochina, which is also a very diverse place, but they are more centered on these more related niches. So we were thinking, well, maybe these reticulations of the subgenus Curcuma could have some role on the diversification of these lots of species, and they can eventually explore new niches. And with this in mind, we have these questions like, can the reticulation that was already estimated on previous work be supported by biogeographic reconstructions following these premises that I already told about the overlapping? And what about if this reticulation actually enhanced the diversification rates in curcuma? So maybe this is one example of how hybridization in the past could lead to a boost of diversification on this clade. And finally, can we also find other explanations for change in evolutionary rates or the invasion of this new environment? So to tackle these questions, we collected samples from the entire subgenus, from the 5 lineages that spans all from India, Southeast Asia, as I mentioned. And we use around 1,100 target low copy genes and the complete chloroplast genomes of 62 species of curcuma. To do so, we built a maximum likelihood tree that matches the reticulation process that was already found. And just want to mention that actually we found different topologies, like because we are dealing with hybridization here, but this one here in the slide is the most likely that would happen. So we also dated this tree because we are interested to see what happened in the past and when that happened. We use some secondary calibration from the sister Costaceae, which is the sister family of the gingers, and to reconstruct the past, like to reconstruct the biogeographic bears, to reconstruct the biogeography of these plants, we use biogeobears, which is our already classical way to deal with biogeographic reconstruction, using 7 bioregions that we follow this work here from 2015 that basically differentiate every sub-region based on floristics, composition, also some environmental variables. And here with the history of curcuma. So around 14 million years old, this is the orange of the clade. So somewhere here, so as you can see here in the first pie chart, sorry, in the first pie chart, we see that the orange of the clade is somewhere here in this Siamese region, or a combination between Siamese of East Cambodia, but for sure somewhere here in Indochina. And after that, after like 2 million years, we see the first divergence of the clade, still somewhere here in Indochina, and we see the diversification of the 2 main clades here in Pura Necomata and the other ones. And now here comes the interesting part, because around 10 to 8 billion years ago, we see the reticulation process happening here. And we can see that the reticulation happens somewhere here in the Indochina region. We don't have 100% sure which of these bioregions, but for sure we know that they are some kind of overlapping spatially. So at least for this first prerogative, the reticulation happened at the same place or neighbor areas, or in a combination of these areas here. Sorry again. And also, when we are thinking about time, around 10, I know this is a very large period of time, like 2 million years, but there are some overlapping between the divergency of the ancestrals of here, Roskeana, and the divergency here of the ancestrals of the entire curcuma. So this is a really cool way, I think, to show how reticulations happen in the past, both overlapping spatially, but also temporally. And keep following the biogeobears, we see here that after diversification, after diversification, here around 10 million, we see that curcuma invaded these new areas. Like there are some still curcuma, subgenus curcuma here in the Indochina, here in Malaysia, but they are the only lineages that occurs here in Bangladesh, also here in Northeast Asian, even here close to the Himalayas, like in Nepal, but also around here in these areas on West India and also in Sri Lanka. So this is a really good way to think, wow, this plant actually reach new places after this diversification. And that's why we're interested to see, well, maybe the reticulation fuel the diversification of this clade. So I was trying to see this using like BAM to check for any reticulation difference between any diversification rate difference. But here in the black arrow, I couldn't find any difference. Actually, the diversification accelerations are more common in the past, way before the reticulation. So at least with this kind of analysis, we couldn't find a different rate of diversification after the ancient reticulation. So, but the niches of these plants are really different, are really divergent. So we were trying to explore, well, maybe some other phenomena help diversify this clade. So I built the niches following the 19 classic bioclimatics and did some ecological overlapping to see if the niches of these plants are somehow overlapping. As you can see here in the first column, the niches of Curcuma are really, really different from the other. Like there's almost no overlapping. This is like the probability of overlapping between niches, and they are really slow for comparing to the other counterparts. So, well, the niches are really different then. So what I did, I estimated that I calculated some correlation between ancestral temperature from like 15 million years old with diversification of this plant. And I found actually that there are some divergence, some evolutionary rate changing when I correlated this with environmental, especially low temperature. So this is where I am still working on this project, because this is like from maybe last month that I found this result. But what about the Himalayas? What about the monsoon regimes, which are really important drivers of diversification on these regions? So this is the next steps of my work, to see if it's not the reticulation that boosts diversification on this clade, what was, right? I want to see if maybe it was the different climate changes at that time, around 8 to 10 million years old. So the take-home messages here that I want to show you. If you have any reticulation in your group, maybe you can use biogeography reconstructions to try to evaluate it or use it as a secondary line of evidence to support that reticulation or not support. And as Eliska already showed you, and also Clarice showed you during the morning, these hybridizations can actually fuel the diversification rates, although here I couldn't find it. But more analysis, specifically for my own work, is needed to disentangle the role of other variables like climate changes and mountain uplift. And that I would like to thank our colleagues and also the funding for explore all this very nice group of plants and to be here, and also the botanical gardens that keep our plants alive. So thank you.
Questions?
Thank you. Very nice work highlighting the role of biogeography. My question is about fitting these models when you and others clearly showed that there has been introgression in the past, so the tree is reticulate and not bifurcating. How did you account for that? Because biogeobears and any comparative method, most comparative methods, assume a bifurcating tree.
Yeah, so that's why when we are reconstructing the tree for the biogeobears, we are trying to use different topologies. So because it's a reticulation, so the topology will change eventually. So this the tree that I showed you here is the most likely to is the most congruent of the ones that we produced. But for sure, when we are dealing with this kind of stuff, like in the paper that we are eventually we publish, I need to put the other trees as well. So you need to have some room to some incongruence between trees.
Any other question? Well, thank you very much to Marco for his presentation.